Okay, very good. Thank you, David. Let's press a button here, and with luck, we're off. So Karen and I decided to switch order of our talks. I'm going to talk in the concrete about what she showed you sort of in the abstract, show you how all those little boxes that she talked about were actually checked. The title of the talk is Targeted Therapies for Pediatric Low-Grade Astrocytomas, PLGAs. I want to talk to you about 4 special challenges in drug development for pediatric gliomas. I think challenges which are kind of generic in nature, not just for low-grade, but for all. And as a scientific vehicle, like I say, I'm going to talk to you about development of a brain-penetrant drug for BRAF mutant PLGAs, which is actually going into clinical trials, a trial that Karen discussed for you just a minute ago. A quick refresher of where we've just been, stuff that Karen showed you, also Ramin and Keith. Low-grade gliomas in kids are a BRAF disease. Activating mutations in BRAF seen within about 2/3 of all PLGAs. They come in 2 flavors. One is the canonical V600E point mutation first discovered in adult melanoma. There are 2 small molecule RAF inhibitors, FDA approved for this indication. Picture of them here. Point to dwell upon here just for a second is both of these FDA-approved RAF antagonists for melanoma are type 1 inhibitors that bind the active or DFGE in state of the protein kinase. Unfortunately, the common mutation, not exclusive, but the common mutation in PLGA is a truncation fusion protein that's unresponsive to type 1 antagonists. What you do with this truncation fusion event is you whop off the autoinhibitory amino terminus of BRAF, leaving a catalytically active domain, but it cannot be autoinhibited. It's active in the absence of active RAS signaling. This is kind of a pie chart of how the mutants are distributed in the cluster of low-grade astrocytomas that Keith Ligon talked to you about. The pilocytic astrocytomas, JPAs, are the most common form, WHO grade 1. And you can see here that this truncation fusion event is almost diagnostic for juvenile pilocytic astrocytomas. 85–90% have this mutation. If you go to the somewhat higher, WHO grade 2 groups of the low-grade astrocytomas, low-grade gliomas not otherwise specified, the ganglia gliomas, the pleomorphic xanthoastrocytomas, now you begin to see occasionally the V600E point mutation popping up, together with the truncation fusion on a mutually exclusive basis, as you might expect, okay? And the reason I show you this is that the type 1 inhibitors approved for melanoma work on the V600E mutation because it alone functions as a monomer, but the KIAA 1549 BRAF oncoprotein functions as a dimer, and the melanoma drugs do not work. In fact, as I'll show you in a minute, they can be counterproductive, and Karen touched upon this actually in her talk. There are other problems with the type 1 RAF inhibitors for melanoma. One is that the melanoma patients treated with these drugs develop skin lesions, keratocarcinomas, and adenocarcinomas, and this is due to the paradoxical activation effect that the type 1 inhibitors have on wild-type RAF proteins. And the other problem that we'll get to in just a minute is that the blood-brain barrier penetrance of these drugs, vemurafenib and dabrafenib, is quite poor. So, against that backdrop, we set out to develop or discover RAF inhibitors that would access the blood-brain barrier and work on both flavors of BRAF mutants that we do see in the low-grade astrocytomas. And there are 4 special challenges that we had to overcome. One is cell lines or lack thereof. 2 is mouse models or lack thereof. 3 is blood-brain barrier. And 4 is, as Karen touched upon, the MTAs. So the cell lines, put quite simply, the problem is that low-grade astrocytomas cannot be passaged in cell culture or in immune-suppressed mice. There's a fellow named David James, started off at the Mayo Clinic, went to UCSF, has spent 30 years trying to put human gliomas, astrocytomas into cell culture or immune-suppressed mice. In 30 years, he has never been able to propagate anything lower than a grade 3 glioma astrocytoma in culture or in mice. Just can't be done. And there are no useful mouse genetic models. Indeed, if you had a true genetic model for a pediatric low-grade astrocytoma, as you'll see in a minute, the tumors would take about a year to develop, and it'd be absolutely worthless for drug screening, drug development. So to deal with both these problems, we built our own mouse models for cell lines. And the point of departure for this is the observation that pediatric low-grade astrocytomas are not astrocytomas. I think Keith touched upon this. We're kind of stuck with a name as a historical artifact. 50 years ago, it was thought that the only replication-competent cell in the brain was the astrocyte. So since a tumor has to initiate in a replication-competent cell, they must be astrocytomas. We've since discovered the postnatal brain stem cells that are much better candidates for cell of origin. Here we show you from Keith's lab immunohistochemistry of a WHO grade 1 and WHO grade 2 low-grade astrocytomas. These guys are just chock full of the OLIG2, BHLH transcription factor OLIG2, which is a neural progenitor cell marker. These tumors in kids are not astrocytomas, they're neural progenitor or neural stem cell omas. So that was the point of departure to develop cell lines for drug screening. We validated 3 mouse models. The thing we do here is start with a wild-type mouse or INK4A ARF or P53 conditional null. We'll talk about these in a minute. Take neural progenitors from the mouse embryos, put them in culture, use retrovirus to install either the point mutation or the truncation fusion mutation. Come in 3 flavors. The P53 axis in pediatric low-grade astrocytomas is almost always unperturbed. None of these cells are P53 mutant, none of the tumors are. Some of the WHO grade 2s lose INK4A ARF. So what we did was, for the juvenile pilocytic model, took completely wild-type neural progenitor cells, put in the truncation fusion mutant. As you see here, they're semi-tumorigenic. We'll get to this in a minute. To model the WHO grade 2 type low-grade astrocytomas, slightly more aggressive, we took INK4A ARF neural progenitors, put in the V600E mutation. You see they do a little bit better. And because for drug development, you like to get your bad news fast, we made a very aggressive, what we call pathway-relevant model cell line, taking P53 null neural progenitors, genetically unfaithful to the disease. And here you put in either the point mutation or the KIAA. And these are, as you can see, quite aggressive. This is the actual data. That's a juvenile pilocytic mouse model. It makes tumors, but it takes almost a year. Really can't do anything with it. This is our WHO grade 2 low-grade glioma, not otherwise specified model. Makes tumors in a reasonable period of time. You could work with it. But here is our pathway-relevant model, very aggressive. The V600E mutation kills all the mice within 3 weeks, and the truncation fusion event still comes on in a timely way. Tumors coming up in a couple of months, mice starting to die. We call it pathway-relevant because if you put in vector alone or wild-type BRAF, there's no tumors. So although it's P53 null, it's addicted to oncoprotein. You have to have BRAF to make the tumor. Another problem, we touched upon this briefly, the blood-brain barrier. There are those, not Tyler Jacks, who say it's not important. And it really is. And in fact, you can see here a kind of a perverse disconnect with type 1 RAF inhibitors and the blood-brain barrier. It is true that with gliomas, on occasion, there is local breakdown of the blood-brain barrier. In fact, with the type 1 tumors, the juvenile pilocytic astrocytomas, it's rather common. Here you see contrast enhancement. There's been local breakdown of the blood-brain barrier. Perversely, though, in these tumors, where the drug could presumably get to the tumor, these are the ones characterized by that truncation fusion mutation where the drugs don't work. Dabrafenib, not going to touch these things. For the somewhat more aggressive grade 2 astrocytomas, the more infiltrative, these, as I showed you a minute ago, sometimes do come up with the V600E point mutation where the drug could work, but more often than not, because they're infiltrative, they have an intact blood-brain barrier, the drug can't get to them. Karen did show you one response in a V600E mutant glioma, but I think what she didn't mention is that was a contrast-enhancing tumor. So you're going to have troubles with those. So how do we image the blood-brain barrier? I show you here some technology developed and really refined to an art by Natalie Agar and her colleagues at the Brigham and Women's. Multi-mass spectroscopy. What you do here is you take a mouse and gavage it with your favorite drug, or a rat, or a dog, or if it's a patient, you have the patient swallow a pill. And then at some time, if you're choosing, if it's a mouse, you chop the head off, excise the brain. If it's a patient, you start your awake surgery, remove the tumor. Make a cryostat frozen section and coat with the matrix for multi-mass spectroscopy. Rasterize this frozen section in an XY axis with a laser dialed down to a tight diameter, 25 microns. Each little puff, you get size fractionation on the mass spec tube. You presumably know the molecular weight of your favorite drug, so you can pick out the peak and assign a color to it. And you also would like to, while you're puffing away here, find something that you know cannot possibly escape the lumen of the blood-brain barrier. Initially, we used an impermeable dye, we meaning Natalie. More often now, we just use heme, which has to stay within the blood-brain barrier. And so colorimetrically, if your colors segregate, if it's Christmas, then the drug is escaping the lumen of a capillary and accessing the interstitial spaces of the brain. If they overlap, you got Halloween, the drug is stuck inside the capillary, can't get out, okay? And this is Natalie, who's responsible for all this work. This is just kind of what it looks like. This is a nice picture of a drug, BCAM120, which is a small molecule PI3 kinase inhibitor with good access to the brain. You can actually get the time course. This is 1 hour, 2 hours, 4 hours after gavaging the mouse. So you can see the drug starting to come out. Here's the heme right here. The drug is in green. Other things you can see with this technology, you can actually see regional differences in stringency of the blood-brain barrier. Here, Natalie and her students gavaged a mouse with BCAM120, waited a few hours, and then sectioned the entire brain from front to back. And as you move from front to back, you can see, getting in the middle of the brain towards the ventricles, you can see drug appearing, and then somehow there's less drug as you go further back. So, last thing you need to deal with here is the big pharmaceutical houses. Low-grade glioma is an infrequent enough disease. I'm really not interested in tool compounds or entry-level drug screens. I like to work with something that at least looks like a drug. It's been tested. We got an LD50 in rats. It has a reasonable half-life in blood. But to do this with the big pharma, you're dealing with lawyers. You pick out one drug of interest, your lawyers talk to theirs, you wait a year and a half, maybe you win, sometimes you don't, sometimes it's your own lawyers that are the problem, not theirs. And God help you if you want to try drug combination or compare the drug from company A to drug from company B. Companies don't like their drugs compared to each other, they hate drug combos that don't involve it. So if it's a real drug, the companies have composition of matter protection on it already, the structure's been published. Just go to your medicinal chemistry core, and the Dana-Farber Harvard Cancer Center has one, an excellent one, directed by Nathaniel Gray and Sarah Burlage. Tell them your problem, and they will reach out to a contract research organization in India or China. A few $1,000, they'll whip up a half a kilo, and you're off to the races. So you can do anything you want with it except put it in patients, right? Skip the lawyers. So having done that, here's a panel of small molecule RAF inhibitors that our core gave to us. And I draw your attention to these two guys right here, which in contradistinction to debrafenib and vemurafenib, which are type 1 inhibitors, these are type 2 inhibitors that bind to the allosteric site of the kinase, the catalytically inactive or DFG out form of the drug. And we're off to the races. Let's discover a brain-penetrant RAF inhibitor. So this is the pathway-relevant cell line that I talked to you about a minute ago, transduced either with the V600E or the truncation fusion mutant, treated with debrafenib or vemurafenib, the two type 1 inhibitors, and you can see that the V600E mutant responds very well. The truncation fusion cells are not responding well at all. This is the Western blot of phospho-ERK, the surrogate reporter for BRAF activation. You can see we're suppressing the V600E mutation, not touching the truncation fusion. In fact, here you see you're getting this paradoxical activation event that the type 1 inhibitors do. Bad news for kids. Here are the pair of the type 2 inhibitors we got from our medicinal chemistry core, efficiently hitting both the V600E and the truncation fusion. None of this paradoxical activation business. Everybody's shut off. One of these drugs, MLN2480, which has had a name change now, is Karen, it's Takane drug now, 5480, something like that. Takana's acquired Millennium. One of these drugs has very nice blood-brain penetrance. This is a H&E section of a mouse injected with this pathway-relevant tumor. This is healthy brain over here. Here is the heme. Here is the drug over the region of the tumor. You can see segregation. Those of you close to the screen, actually I blow it up here. You can see we're even getting drug into the healthy areas of the brain, and clearly the drug is segregating away from the heme inside the tumor. This just compares the penetrance of MLN 2480 to dabrafenib. You can see a big difference in brain penetrance there. The drug works. It suppresses the growth of both V600E and the truncation fusion mouse tumor line in the mouse brain. We mentioned that it's hard, basically impossible, to propagate these juvenile pilocytic low-grade gliomas in mice or in culture, but you can do short-term things with them. So here's a 2-hour experiment on cover slips. We got freshly isolated cells from Keith Ligon, and that's something he mentioned he can provide for you. He can't give you cell lines, but you can get the cells from them if, on occasion when there's a little bit left over. Here is phospho-ERK. Because these are freshly isolated tumors, not everything on the cover slip here is a tumor cell. You got fibroblasts, capillaries, but you can use OLIG-2 as a marker for the actual tumor cells. Here's vector-controlled DMSO. Here's a Millennium drug, shuts off the phospho-ERK. Here's a vemurafenib, not touching it. That's a short-term experiment. We can do a little bit better than that. Rosalind Siegel and her colleagues at Dana-Farber have developed basically an avatar clinical trial method. You take thin sliced sections of a 2-week-old mouse, and you can overlay them with your favorite tumor cell type, and you perfuse these cells with a red fluorescent dye. You can see here's what the slices look like at day 1. 7 days later, there's been growth here, as you can see with DMSO. If you treat with a Millennium drug, you can see, I think, much less growth. Here with the vemurafenib, looks like, if anything, a little bit more. This is just quantification of what I just showed you. These are our mouse models. This is the truncation fusion of DMSO with the Millennium drug and with vemurafenib. And here's the same with the human juvenile pilocytic astrocytomas with the BRAF duplication. So, wrap it up. This kind of summarizes, I think, the cluster of the morning talks. I'm looking forward to hearing from the engineers right now, but what all of you have sort of heard so far is that primary cancers of the brain have now surpassed leukemia as #1 cause of cancer-related death. Astrocytomas, aka gliomas, as Keith tells us, the most common form of pediatric brain tumor. The pediatric and adult gliomas, as I tried to push, are completely different diseases, completely different one from the other. I ticked off a number of different ways they're different. For pediatric glioma drug development, a few thoughts. One is don't count on pediatric cell lines. For the low grades, they're impossible. Even for the high grades, Keith has had a few successes, but these tumors are infrequent. You're not going to get something like the cell line encyclopedia that the Broad Institute and Novartis have developed, 500 different gliomas, where you can really get strength from numbers. I think pathway-relevant murine models are superior for entry-level drug screens in many respects. You can make isogenic drug comparisons. Contrary to popular rumor, the blood-brain barrier is important, but there's ways to deal with it. And I think a medicinal chemistry core is key. Don't wait around for lawyers. So these are the people, many of whose names you've already seen here. Postdoc Grace Sun in my lab did all the cell biology, and the other names I think I mentioned in passing. Much thanks to the Pediatric Low-Grade Astrocytoma Foundation that funded a lot of this work here. Thank you much.